extension here, or and we'll go ahead and get started on that.
Thank you. Good afternoon, everybody. So I'm Olga. I'm finishing my PhD in Washington State University. I work with Dr. Tobin Peaver, and my main projects are Botrytis on red raspberry. So today I'm going to talk about a couple of projects that we've been working on Botrytis gray mold in red raspberry here in Washington State over the last 4 years. So first, I would like to start why we are talking about Botrytis so much in here, and why Botrytis is a problem in Western Washington. Well, the fungus loves the weather of Pacific Northwest. From over 60 years of research, we know that the pathogen prefers cool, wet environment, and infects, and in these conditions, infects plants very rapidly and spreads very effectively. And the optimum temperature for Botrytis infection is between 15 and 25 Celsius, which is probably similar to what we have here in Western Washington. Presence of free water, which is rainfall, or even high humidity can facilitate an infection. So it's not surprising that Botrytis fruit rot is the main problem in raspberry production here in Western Washington. I would also like to talk about already gained knowledge how Botrytis infects red raspberry. So what do we know about the infection process? The main hypothesis that we have right now, that was established about 50 years ago, that fungus infect raspberry flowers that you can see here through its female part, carpel, and doesn't show any symptoms on flowers. So stays latent in the flowers until fruit starts developing and maturing from these flowers. And then on fruit, we can see pretty distinct symptoms of gray mold. And based on this hypothesis, the main management strategies here in Washington and all over the world are built on. So here is a quote from Pacific Northwest Plant Disease Management Handbook that suggests growers to start spraying first at 5% bloom as soon as flowers get open, since we think that fungus infects flowers and stays in flowers until fruit ripening. And repeat spraying every 7-10 days depending on weather, especially pre-harvest and especially if the weather is wet, since we know that disease is very conducive by wet weather. That's what growers do here. So all the disease management strategies are built on a calendar basis, and they start spraying from the time when flowers open all the way through the harvest. However, this hasn't been, there's not much research data on testing different fungicide programs time-wise, depending when to start spraying in raspberry system. One of the research that we were able to find in the literature was from Ohio State University, where they actually compared 3 types of spray programs, including full seasons that you can see here. Another one was sprays were restricted only to bloom period of red raspberry. And the third one, the plants were sprayed only pre-harvest. As a result, in the result of their 2-year study, when they looked at the disease incidence, they could see that in the program where the sprays were focused only on bloom, the disease incidence of Botrytis gray mold was the highest, comparing to full season and pre-harvest that actually didn't really differ between each other. So this gave us some food to think about, that how important our flowers actually are for the botrytis infection of red raspberries? And is there anything that can be or should be changed in the spray programs that currently applied? As I mentioned before, we started working on this problem from 2015, and here I just briefly going to show you previous findings from 3 years of our research. So in order to see at which developmental stage fungus infects plants during the raspberry season, we divided the whole process into 7 developmental stages. So starting from close green buds all the way to the ripe fruit. And 2015 and 2016 summers, on a weekly basis, we kept coming to this field and kept sampling all these stages that were present on the field and seeing in how many samples there was botrytis. Surprisingly, what we saw that the level of colonization of flowers was very low comparing to the colonization of fruit. And we see that with the flower and fruit development and maturation, percentage of colonization of these samples was increasing. And this data was consistent over already 3 years. Since I mentioned that wet weather is important for the disease development, we also looked at the rainfall data. So let's focus first on this 2015, 16, and 17 years data. This is accumulated rain from the beginning of April till the middle of August of these 3 years. So we see that during flowering in 2015 and 2016, there was not as much rain as comparing to 2017, when the amount of accumulated rain was higher during flowering season. Again, if we think about main hypothesis that fungus is thought to infect flowers, so this rain must have facilitated in better infection of flowers. However, in our data, we didn't see the increase of flower infection even when it was more rainy than previous years. If we look at this black line, so this is rainfall data from 2012 year. So we haven't worked on Botrytis gray mold that year, but what we know from growers is that that was probably last bad Botrytis year here in Western Washington. And when we looked at the rainfall data, we can see that the amount of rain during the flowering wasn't very different from, for example, 2017, but it was much more rain during fruit development and harvest. That makes us think that this additional rainfall during harvest may actually induce infection of fruit directly. And seems to be pretty important since gray mold this year was pretty bad here in Western Washington. I'm gonna switch to the project that we worked in 2017, 2018. Since we didn't see a lot of botrytis in flowers, we decided to test how fungicide sprays protect these flowers. So we tested the effect of standard fungicide program that is used by growers here on botrytis colonization of red raspberry. And we also wanted to test, do floral infections lead to fruit infections in raspberry fields? So we compared botrytis isolated colonized flowers to that colonized fruit using genetic markers, just to get to use a different technique in order to test the same hypothesis that we were working on before, using more precise markers. This is the experimental design of the project that we did in 2017 and 2018. So we picked 2 fields on commercial farm here in Whatcom County, and each field had plot which was left non-sprayed with fungicides at all, and the second plot was sprayed with fungicides as a regular program as all other fields get sprayed. The only difference between these fields was the age of plants. So field number 1 was younger comparing to field number 2. In this experiment, we decided to focus only on 3 stages of raspberry flowers in fruit, not all 7. So we picked fertilized flowers or late flowers, so flowers that start becoming fruit, green fruit, and ripe fruit. So we went to these 4 plots over 2 summers and sampled these 3 stages and checked for how much botrytis is in these stages. If we look at the effect of fungicide applications on botrytis colonization, again, on the x-axis, our 3 stages, on the y-axis, percentage of samples that was colonized for each of the stage. What we see, first of all, about the same amount botrytis colonization of the late flowers comparing to previous years. But also what is important, we didn't see any difference in colonization of the flowers comparing sprayed versus non-sprayed plot. When we looked at the colonization of green fruit and ripe fruit, it was again consistently higher comparing to flowers, and they were statistically significantly different, sprayed versus non-sprayed. But again, if you look at the sprayed plot, about 80% of green fruit and 90% of ripe fruit had botrytis in them, even on sprayed plots. Again, and this data does not support the hypothesis that flowers are most important route for the fungus to cause disease on raspberry plants. If we look at the colonization of flower parts by botrytis, so we dissected flowers into this female part carpel, sepal, and stamen, and looked in which organ there was the most amount of botrytis among those flowers that had fungus inside them. And our data was consistent with previous findings in research and previous findings during 2015 and 16 years. So if there was fungus in flowers, it was mostly in female part carpel and much less in other parts. But also, it didn't differ between sprayed versus non-sprayed plots. If you look at colonization of ripe fruit by botrytis, and here I am showing data for 2 fields separately, and in a second I'm going to explain why. So we looked at different morphological part of fruit to see at what part we had the most amount of botrytis and if it differed between sprayed versus non-sprayed plot. So our druplet, which is fruit itself, which is in the most interest of growers because that's what we harvest and sell, while stamen, sepal are receptacle as well. So in field number 1, if we look at the percent of samples that were colonized at each of these morphological parts, we see that first, fruit itself, almost every fruit had botrytis in it. If we look at the field number 2, we can see a little bit different picture. So the non-sprayed plot, 100% of fruit had botrytis in it. But the sprayed plot, only 30% of fruit had botrytis in it. Why this difference can be so big between these 2 plots? We speculate that the reason is the age of the plants. Since the field number 1 was much younger, it had much more dense canopy on plants during those years. Comparing to the field that was older, it had less leaves and less vigorous growth. So maybe that affected the efficiency of fungicide coverage of plants. So that's something that it can be, again, it can be checked and studied more, but something that can be also taken into account, that if age of plants and age of fields, on young fields, there probably will be more disease and infection just because we cannot really protect flowers and fruits that are behind the leaves. If we look at this data in a little bit different way, so here we look at quantitative data, average number of botrytis colonies per ripe fruit. Because as I said before, when we looked at flowers, if flower was infected with the fungus, there was usually just 1 colony per flower. So only from 1 part of the flower, the fungus grew. But when we looked at the number of Botrytis colonies per ripe fruit, again, field number 1 and field number 2 separately here for the same reason, we see that in unsprayed plot, in fruit itself, the average number of colonies, so places from which fungus grew from one fruit, was about 6 from field number 1, while on sprayed plot, it was a little bit more than 3 average. If you look at the field number 2, the difference even bigger. So the number of colonies, average number of colonies per ripe fruit in non-sprayed field was again around 6, while comparing to sprayed was about 2. So what it tells you, this data shows us that fungicide applications seem to be effective at the fruit stage, even though we didn't see that effect at flower stage. So they do protect fruit, maybe not from infection, but at least from growing, from fungus growing out of the fruit. So this data shows that sprays are effective on ripe fruit stage. So over this few years, what we saw low rates of colonization of raspberry flowers. Botrytis colonization increases as fruit develops. And fungicide sprays didn't affect Botrytis colonization of flowers. That's what we've seen over these 3 years. And this led us to our second project to work on, to compare fungus in flowers and fruit using genetic markers. Because since it's a microscopic organism, you can't really take 2 individuals and put them next to each other and look at them and see are they different or no. But using molecular markers is much easier to do. For this, we asked the question, are fruit infections a result of flower infections? And we considered 2 scenarios. Scenario number 1, that fungus infects only flowers. And scenario number 2, that fungus infects multiple stages of red raspberry. So let's think about the scenario number 1. If fungus infects only flowers, so let's imagine on a field we have some genetic diversity of the fungus, and each of this arrow represents different genetic type, colors of their arrow. So for example, flowers get infected by two genetic types, black and red. What we expect to see in fruit if consider this scenario? Well, we expect to see the same genetic types in fruit if we think that all the infection came from flowers. Scenario number 2, what if fungus infects multiple stages? So we have some genetic diversity, subgenetic types that infect flowers. Then later in the season, we have the same genetic types that infect flowers, but also some new genetic types. Here is, for example, green arrows that infect fruit come later to the field. And on a ripe fruit stage, we have even more diversity. More genetic types came to the field and infected our fruit. When we looked at our data, what we saw? First, on the x-axis, 3 stages that we looked at. And on the y-axis, average number per inflorescence of colonies of Botrytis and pathogen diversity, the genetic types. Because for this project, what we did, we picked 20 inflorescences, marked 20 inflorescences on each of 2 fields. And we first sampled 1 flower from this inflorescence. Then later in the season, we came back and we sampled ripe fruit from this inflorescence. And later we sampled green fruit and ripe fruit from this inflorescence. Again, asking a question, if all the infection comes from flowers, then all of these genetic types should be similar, should be the same in inflorescences. But with this data, we didn't find the evidence of such case, because the number of colonies, again, increased with fruit development, and also pathogen diversity, so number of genetic types increased with fruit development and maturation, and was the highest at ripe fruit stage. What would we expect to see if scenario number one, when only flowers get infected, would be the case. Probably that the genetic types that we found, again, in flowers would be the same that we found in fruit, or at least there would be overlap of the genetic types. But when we looked at this data, we saw that, so this yellow circle represents the, like a pool of genetic types that we found in flowers. And they didn't overlap with either green or ripe fruit genetic types. This area between green and ripe fruit that overlaps a little bit, it shows the amount of genetic types that was found in green and ripe fruit. So about 1% of the genetic types was shared between green and ripe fruit stage, but majority of genetic types in ripe fruit was different from the other stages. Again, it gives, it makes us think that fruit may be getting infected additionally later in the season, but not through flowers. So in the conclusion of this work, what we found, fungicides applications do not decrease Botrytis colonization of flowers, either by incidence or quantitatively, but decrease amount of Botrytis cinerea in ripe fruit, so they're effective on ripe fruit. Diversity of Botrytis increases as fruit matures. Different types colonized flowers, green fruit, and ripe fruit in the same inflorescence, and that suggests independent infection events throughout the season. If we come back to our fungicide spray programs, what it means. So if sprays do not protect flowers and are not affected on flowers, maybe we could reduce amount of sprays that are applied on raspberry fields. And what it would mean for the industry? Well, first, it would save money because you don't need to spend as much money for fungicides. But one of the most important problems, it would decrease fungicide resistance. A very big problem that growers have right now, that because we spray so much, these fungicides lose their efficiency because pathogen gets resistant to it. So if we decrease amount of sprays, it will be the best strategy to cope, to manage the fungicide resistance. Of course, for this, we would need to do more studies and to do more field trials, because we also know that these last few years were pretty dry and not very conducive for the disease. But as for now, with this climate, that's the picture that we've got. I also wanted briefly to introduce another project that kind of this data led us to start working it. Since every raspberry fruit is infected by at least one colony of Botrytis, but we don't know the quantity, and we don't know when it starts affecting quality. So based on this information, we decided to start a project to quantification of Botrytis in processed raspberry fruit, since majority of fruit that is grown here is grown for processing. And that's a project we started working this year. So the current method that is used in processing facilities is hovered mold counting. When you smash raspberry fruit and put it under a microscope and try to find mycelia hyphae in order to quantify how many of them are there. There are a few limitations of this method, however. First of all, you really need to know what you're looking at, and you need to be able to recognize is it the mycelial hyphae or of the fungus, or is it the hair of the fruit. And also, you can't really distinguish between mycelium of other fungi, not just Botrytis. So it's not as precise. Because of that, we propose to start working with this project using Botrytis-specific medium that we use in previous projects, and also quantification of Botrytis amount in macerated raspberry fruit using molecular method qPCR. So we don't have any data on this project, but we just started working on it. And hopefully next year we will be able to share results with you. So that's the project design that we are going to use. We are using field samples that go to processing plant, and the same samples will be used for hover mold, for culture non-selective medium, and the qPCR. And then we'll be able to compare the data from all these three projects together. There are a few more projects that we are going to start working next year, since we got Washington Specialty Crop Block Grant to work on novel diseases, novel disease control strategies for berry growers here in Washington. And these projects will be focused more on monitoring and management of fungicide resistance. Since, as I said before, this is a very big problem, and we need to develop high throughput methods that will help to detect fungicide resistance, their types, and how we can decrease the amount of fungicide resistance. With that, I would like to thank you, all of you, small fruit pathology team from WSU, red raspberry growers that participated in research, and of course, funding sources. Thank you.
We've got time for a quick question. Anyone? Okay. Thanks.
Thank you.
Thank you, Olga.
Thank you, Olga. We have a couple minutes to go ahead and set up for the next presentation. Again, just as kind of a reminder, at the end of this presentation, you will have the opportunity to sign up for pesticide credits, and those sheets will be over on these tables over here, depending on what state you're from, province. Also to remind you, after this session, there will be a social hour, happy hour. We want to definitely encourage you to network with not only some of the vendors, but also some of the just mingle with all the other vendors that are here today.
We're good to go. Go ahead and you can go ahead. You can go ahead and introduce her. You can go ahead and introduce her.
So I'd like to go ahead and our next presenter come on up and perhaps get started. Again, Bo Heisel, regional extension specialist, is going to be presenting next.
And she's actually coming in remotely. So we, as you all probably are aware at this point, we live stream this to Prosser in southwestern Washington, and Gwen's going to actually present remotely. So Gwen, if you can hear us, go ahead and let's check the mic. I've seen your screen.
Yeah, can you hear me? I think I can hear myself through yours. So Chris, just let me know if I can start.
Go ahead. You're a little bit echoey, but we'll make it work.
Okay. It might be. All right. Okay, so thank you very much for letting me present from Prosser because, well, there's a whole group of growers here that are here all day. So I want to present to you on research that we did that was funded by the Oregon Blueberry Commission and done with collaboration with Juan and Starhawk, who's our graduate student at Oregon State University, and some graduate students from WSU. And so this is an evaluation of 3 sprayers that we use commonly used in blueberries. And if you've heard me in my talks before, my motto is every drop to the crop. So that's our goal in spring. And sprayers, the 3 things that have the greatest influence on any sprayer, including these 3, are droplet size, air, and weather. And I'm going to go through each one of those and show you how these sprayers will be adjusted or adapted and how it plays into getting every drop to the crop. So, just tap it once. Okay, just hit next with your arrow. So, there is no perfect machine. So, all can be operated poorly or perfectly. So, I'll put that there, that even if you think, man, this machine is really recommending this, I'm not recommending a machine, then I've really seen them get screwed up. You don't really provide good coverage. Okay. So my assumption when you're doing this is that you have maintained your sprayer properly. That includes all these things you normally do. But if you don't do these, some of the sprayers we're going to talk about can have a lot of problems. Okay. So the sprayers that we evaluated were a pack blast, a Canon sprayer, which is the second sprayer being shown, and an on target. And the pack blast you're probably the most familiar with, the small version of a rear air blast. 5 nozzles on each side, all has smaller fans. The Canon sprayer is marketed in their words that
Gwen, can you hear me? Gwen, can you hear me? Can you please come closer to the mic? Maybe she doesn't hear me.
Yeah, I hear you.
Okay, can you just get closer to the mic? It's really echoey.
So you know what I'm going to do is I'm going to get off stage and go closer.
That's fine, for the record.
Okay, is that good? Or better? Okay, Chris, is that any better? All right, I'm just going to keep talking, and you can text me or tell me if it's not. So there are 3 unique sprayers, and okay, so the Canon sprayer is marketed so that
Apologies for this, everyone just hang tight.
hey, what's up? I'm standing right next to my microphone and right next to the receiver.
Yeah, just be careful of feedback. Gwen, can you please get closer to the mic? We're not getting anything from you at all.
Chris,
Okay.
I'm standing, can you hear me right now? Chris, I'm not near a mic. The mic is a portable, like I'm not, like it's right at my mouth.
That's actually working really well. Do whatever you're doing right now.
Right now?
Yeah.
Okay. All right. Good. Okay. So the Canon sprayer is advertised as a sprayer that will, you can drive it around your block if you've planted too close or you don't want to drive through, and it will actually spray in a 100-foot swath all the way across your field. So then the last one, which is all the way on the right, the white tubes, that is an on-target electrostatic sprayer. It runs by having a weak charge on small droplets, very, very small droplets. And it's supposed, that weak charge is supposed to be attracted to the weak charge of a plant surface. So we went through, we tested these at rates that were gallons per acre that were acceptable for each machine. We did adjust for that within the data. And then we looked at each coverage that we're going to talk to you about. With that picture at the bottom with the green dye, that's a dye that fluoresces, and we spray with that and then measure it in the lab to see where and how much material went with each sprayer. Okay, so what we did is we did each sprayer. There were 6 canopy zones, and so that we had 3 going across the top of the canopy, 3 going across the bottom of the canopy, and then we had 3 rows that we measured from the spray row of aerial drift, which we took poles that were 1 foot, 2 foot, 3 foot above the canopy, and then we also measured ground deposition that were 1, 2, and 3 rows apart. So the Canon sprayer, we actually set up to spray 3 rows at a time, a 30-foot swath instead of a 100-foot swath. And so it had a spray row of 3 rows, 30 feet. And then from there, we went over 3 rows with aerial and ground drift. And the other sprayers spray 1 row at a time. Okay, so now you know what we were doing. Let's talk about those sprayers in relationship to droplet size. So droplet size, remember that nozzle type matters, and it dictates the size and whether it can be adjusted or not. And the higher the pressure on the machine, the smaller the droplet size. And so if you are not convinced that you should care about it, here's how labels, regulations, and such are coming down. We have many herbicide labels that are now listing on them the droplet size, whether this one here lists that you have to have a medium droplet size, you cannot use a coarse droplet size. There are some canopy labels, some pesticides that are used for canopies, pests and diseases that are also being considered to be regulated with canopy droplet sizes. Also in Washington, we have our new, well, in the US, we have our new worker protection standard rules. And in there, which you guys are all familiar with, there's the application exclusion zone. And within that, they have a determination that says if you're using an air blast sprayer and it has fine or very fine droplets, you have to have a 100-foot buffer. If you're using a sprayer that can produce coarse or larger droplets, you only have to have a 25-foot buffer. Unfortunately, the way the law is written, even if you put in droplets in your air blast sprayer that produce coarse or larger droplets, they're still going to restrict you to that 100-foot buffer. But that's just because they didn't understand all the regulations, and there's been a lot of questions afterwards. There is a lot of discussion about if you prepare air induction nozzles, other types of technologies that get larger droplets, less drift, could your regulations be a little less severe? Okay, so nozzle types. So now the pack blast sprayer uses the nozzles on the left, of course, that you know. So it can change, any of those. It can change into a disc core. Yeah, hopefully you're using ceramic disc core. It can use one-piece nozzles, which produce a fine, very fine droplet. And it can also use air induction nozzles, which would allow you to use a very coarse, extreme coarse nozzle. The Canon sprayer and other sprayers that are like it use air shear type technology. That droplet size can be very variable, but have a lot of fine to very fine. And the same thing with the electrostatic. It is going to be a very small droplet size on that electrostatic. And the reason being is that the smaller the droplet, the stronger the charge it can hold. So you cannot ever use the electrostatic at high gallons per acre. So 20 gallons per acre is about the max you would use it at. And you need it at a high pressure to produce small droplets, very fine droplets, so it at least holds a charge. If you do anything different than that and get bigger droplets, then you've negated the purpose of the electrostatic. Okay, so a while back, people said, hey, ASABE said, let's make this easy for people. We'll color code it so that everybody can figure out exactly which nozzles to use. So they came up with this color coding system so that if you find red, it means a very fine droplet, orange, a fine, yellow, etc., medium. But when you actually look in a catalog, it's really confusing because they have colors all over the place. It's like a preschool magazine. So the colors that are associated with the categories of the droplet size are actually the ones in the center of the table. So there's a V, C, and a C, and an M. Those are the center of the table. That is the color. On the left side, there's an orange, green, yellow, purple, blue, red bar. Those colors are absolutely arbitrary and actually line up with what the color of those one-piece nozzles or air induction nozzles are. They have absolutely nothing to do with what your droplet size is. So just be careful about that when you're selecting. If you choose to go with the pack blast, which you can change all of these nozzles, and you want to take advantage of droplet size, you're going to end up having to look at it a little carefully. Okay, so let's look at air. So the goal, if you were going to do one thing besides my very first slide, which was make sure all the parts are working correctly, matching the air to the volume of the canopy is the key to proper deposition, or the amount where the spray is going to go. So it's a really simple concept, but it's really difficult to achieve. And so I'm going to show you how each one of these sprayers that we went through, how they performed, and where the deposition was and how it might relate to air. So I don't need you to read all the numbers on the side. This is like a really great graph because it shows you trends on all of these. This is the pack blast. And at the top of all of these slides, at the very top is the canopy deposition. The next slide down is your aerial drift, and your bottom slide is your ground drift. And so at the very top, under canopy deposition, on the left of the gold bar is all the canopy zones that are at the bottom of the canopy. So you're looking at zones that go across the bottom of your blueberry bush. The one to the right of the gold bar that says top, those are the 3 zones that go across the top of your blueberry bush. So it'd be left, middle, right of the top. So what you can see with the pack blast, and we optimized it, we looked at proper deposition, we did our very best, we put up water sensitive paper ahead of time, but the way that axial fan sprayer occurs, we consistently got more deposition in the bottom zones of the canopy than the top zones of the canopy. If you look at aerial drift, in general with the pack blast sprayer, if you look at row 1, which is your second graph in row 1, you get more deposition closer to the row. So this is 1 row over. You get the most drift in the air 1 row from the sprayer. That's good. Through the years, it's decreased as consistently between the second row and the third row. So that's good. Most of your drift stays within 1 row of your field. If you look at ground deposition, it's the same thing with the pack blast because a lot of it is captured in the canopy. 1 row over on the ground is where a lot of that spray goes, which makes sense because if you hit the majority of the bottom of the canopy, the nozzles are putting out a lot more down there, and you're going to end up with ground deposition there, 1 row over, unless 2 and 3 rows over. So now let's look at the Canon sprayer. So in the Canon sprayer, same concept. The canopy is at the top, then the middle is aerial drift, and bottom is ground drift. So the Canon sprayer, remember, is meant to go around your block and shoot sheer all the way across the top of your canopy and then sort of deposit down. It works on a gravity effect. I'm going to spray as far as I can, and then what goes up must come down is what that concept is. So the canopy obviously then has way more deposition at the top and a lot less at the bottom because that's the first place it hits. The aerial drift generally has less deposition further from the sprayer, but if you looked close at those bars, there's often no significant difference between the first row, second row, third row. So what that means is that this sprayer is sort of working the way it's supposed to. It's just that it's blowing all the way across. You're still getting stuff blowing across even 3 rows over. Quite a bit, actually. If you look at ground deposition, it's the same, that you have more 1 row over, but you also get a lot of deposition in the middle of your rows that aren't doing you any good on the canopy. Okay, so now we look at electrostatic. So the canopy if you look at that, it's the most even distribution through the canopy. So if you look between the bottom and the top, there's more similar amounts. The bar graphs are more similarly aligned. The aerial drift, it's even low depositions 1 through 3 from the sprayer, but it's a similar amount, nanograms per centimeter, so it's the same amount as the pack blast. So it's more evenly distributed. It drifts across all 3 rows, but it isn't like it's a huge amount of drift. It's the same amount as a pack blast, but it's a whole lot less than the Canon sprayer 1, 2, and 3 rows over. And ground deposition again is similar, or is low in the second and third row from the spray row, but higher in the first row. Okay, so now this is what you can do for your sprayers. So the summary of that was, of those last 3 graphs, is if the sprayers are working how we would intuitively think about them. You know, if you're intending them to go over the top, you get more deposition in the top. If you are able as a tower sprayer, like the on target is set up where the nozzles go up and down and spray horizontally into the canopy, you get more even distribution between the top and the bottom. But there are still things. You don't have to run out and buy a new sprayer, because there are still things that you can do to adjust your sprayers to make them have greater deposition. This is an example of an extension demonstration that we did in grapes. So those little yellow cards are water sensitive paper. And if you don't have any, you should pick them up, because they're yellow when they start. And then when water hits them, they turn blue. So it's a really great way to look at where canopy spray is going. So what we did for an air blast at the top, and we had this air blast in a grape canopy at full throttle, opened up, and we had all blue, meaning all the spray went towards the top part of the canopy. And we didn't like it. It wasn't even distribution. What we did is that GUTD is we geared up and throttled down, which led us to the bottom picture, where we had more even distribution from all the way on the left, the top square, down to the bottom. All that gear up, throttle down does is it reduces the rotation of your fan. So you're gonna gear up to a higher gear. You're gonna throttle down. So you gear up so you can go the same speed. You throttle down so that the PTO spins at a slower rotation, which means that your fan spins at a slower rotation, and you have more properly matched the volume of the air coming out of the sprayer with the volume of your canopy. It also happens to save you money on gas. So if you have a pack blast or an air blast that you're using in blueberries, looking at it with water sensitive paper, gearing up, throttle down is a great solution. If you have an on target, any other air shear sprayer, a Canon, you can never gear up and throttle down, because that technology is meant so that the droplets are formed when you have high pressure air running through the sprayer, and it will shear the liquid to create lots of fine droplets. Okay, last one. Let's look at weather, right? So temperature and humidity have a drastic effect on droplet size, coverage, drift, and control. And I'm gonna try and convince you of this, because if you have one of these sprayers, you're gonna have to think about how to operate them differently in different conditions. So we have 2 slides left. So temperature and drift in real life. And we didn't have in our blueberry study more extreme temperatures, and we were on the west side, so the humidity was a little bit higher. So this graph I'm showing you here with the, under the big word temperature comes from almost the exact same study except done in grapes and in Eastern Washington. So very often we set a temperature threshold, and on labels it can sometimes be 85 degrees is when you should stop. 85 degrees Fahrenheit is when you should stop spraying. We performed these trials, and the light blue line is the early season trial, and it was at a lower temperature. The dark blue line is the late season trial, and it was hotter and it was drier. So lower humidity, higher temperature. So I want you to look, this is the drift, 1, 2, 1, 2, and 3 rows over. And in the early season, you had very little drift. It was all in one row because your lower temperatures, and we were still lower temperatures, higher humidity. Late season, we went from drift being much higher, 2 and 3 rows over. And the only difference was a matter of from 70 to 84 degrees Fahrenheit and slightly higher humidity. So that temperature can really decrease the droplet sizes before it even reaches the canopy. So now you have to deal with how are you going to use these sprayers properly. So the air blast, if you are spraying the majority of the time in hotter conditions, then you can select a nozzle with larger droplets. I showed you those pictures. The On Target, the Canon, any other air shear sprayer, you can only change your temperature threshold. Because you're already spraying very fine to fine droplets, you want to be really especially careful to be spraying at lower temperatures, below 85 degrees, so that you are not evaporating the droplets before they even hit your canopy. Okay, so then the last thing that I want to show you is that very often we get very warm. Then it starts to grow. Things start to grow really fast. We start to go through spray intervals. We just had our last talk about botrytis or powdery mildew. And are you keeping up with your intervals and how many machines do you need? So there was a great study that was done on, it's a white paper that was done on how can we optimize or the cost savings if we change a spray practice, how many dollars per acre can we save? And the study was done in grapes, but I think it holds true in blueberries and tree fruit. I've shown that to both of those groups. And when it goes down, if you change the top row from 1 to 2 rows being sprayed at any one pass, you can save the most amount of money. Decreasing your spray volume from 80 to 40 gallons per acre would save you the least amount of money. So when you're selecting a machine, if you have something that is like a pack blast, it's hard because that machine will never be able to go over multiple rows at a time, right? But an On Target, we have a 5-row On Target sprayer here in Eastern Washington. So it can go over multiple rows at the same time. So while it has some negatives that you need to be more careful about the droplet size and the temperature and the weather conditions, it has some real pros as well because you could go over multiple rows. The same would be true with the Canon sprayer. The Canon sprayer, while it had a lot of drift, it can spray multiple rows at the same time. The greatest concern by us as entomologists and Bond is that if the majority of your spray is at the top of the canopy, we're worried about SWD in the middle and bottom of your canopy. So you want to make sure that you're getting, when you adjust it, there's multiple heads on it, that you are still getting good coverage in the center and bottom parts of the canopy where SWD can hang out, because it's a little bit less hot down there in the center of your canopy. Okay, so with that, we are at the very end of the day. Chris, I don't know if you can do questions or not, or you can text them.
Are you stretching, Chad, or do you have a question? Any questions for Glenn?
Glenn, can you hear me?
I can hear you.
Okay, we do not have any questions here. Do you have any at your site that you want to handle?
Guys, no, they're good.
Thank you so much, Glenn. That was great. Let's give Glenn a round of
The sound
applause.
got better.
Yeah, it figured itself out. It actually works great. So that concludes. Pesticide credits will be over on the left-hand side of the room. That concludes our like educational content for the day. There's a social hour I would suggest everyone go to. Starts right now. Really good food. Get a drink, hang out, and talk with everyone. And we start again tomorrow at 8 in the morning with the Washington Blueberry Commission. Thank you.
Wait, you want site credits?
Was it?
I don't know what's going on their side, because Joan checked it with me in here. She said, oh, I heard you agree.